Hello. Today, we're starting the first ever session of our class on the Ebola hemorrhagic fever. We are going to be talking about this disease, when it appeared, what the disease was like, and how did it affect the area that it was found in, and the entire world at that. And what we're gonna do is we're gonna follow the chronological order and the events and the perspectives of the book No Time to Lose, A Life in Pursuit of Deadly Viruses, written by the Belgian doctor Paul Piot, who is currently the director of UN AIDS. Not UN for aiding people, but UN for HIV and AIDS. So without further ado, we are going to get started. So our story begins in September of 1976. So the writer of our book here, Peter, he works in Antwerp, Belgium, and he works as a specialist in infectious diseases, and he works in a laboratory that is certified in using centrifuge machines to sample body fluids and diagnose diseases, including yellow fever. And on Tuesday of that September, a package arrived at their laboratory. And this package was sent to the laboratory from Kinshasa. Now, Kinshasa is the capital city of the country that we know now as the Democratic Republic of Congo, and back then what they called Zaire. Basically, it's the same place. And that name, Zaire, is partly why Ebola's full name is called Zaire Ebola virus, because it was found in Zaire. There were other Ebola virus that was found in a different African country, and that was called Ebola Sudan. This one is called Ebola Zaire. So anyway, this package arrived in his lab in Antwerp, and at the time, nobody knew what disease was contained in the package. This was because the package was sent to them with regards to an epidemic that was reported in the DRC, or Zaire, whatever you call it, and it was reported to be yellow fever with hemorrhagic manifestations. So what this means is yellow fever is a type of disease spread around by mosquitoes caused by a type of virus belonging to the arbovirus family. And hemorrhagic manifestations means that there are hemorrhagic symptoms. And what hemorrhagic symptoms mean is that basically eternal bleeding. What this means is that your blood vessels are bursting inside you and they're bleeding into every open cavity you can find, which sounds dangerous and is very dangerous. So as this Peter's lab, right, the lab Peter's working at, was certified for working with yellow fever and diagnosing yellow fever, this blood sample was sent. However, there's a problem, right? In the 1970s, humans were at an age where the advancement of modern medicine was very rapid. With development of antibiotics and vaccines and whatnot, a majority of people at the time believed that pretty much all infectious diseases have been already either identified, or if they have not been identified, would be easily treated and defeated by vaccinations or treatment or cures once they were identified. And especially when it came to yellow fever, a disease that can only be spread when a mosquito bites somebody who's infected with yellow fever, then bites somebody who is healthy. They didn't really think that this sample, which they believed to be yellow fever with hemorrhagic manifestations, was going to be very dangerous or stable. And Peter, sat down with his colleague, a person called Guido van der Groen, who he described as a shy, funny fellow Belgian who is about age 30. Right? And another one of his colleagues, whose name I'm not going to pronounce because this is not English. Sorry. He's Bolivian, right? So they opened it on the lab bench. So how did they open it? They were wearing latex gloves because their boss insisted on wearing latex gloves. They were not wearing hazmat suits. They were not wearing face masks. And of course, they could not fathom at the time that right there in front of them, in the thermos that they were opening so carelessly, was probably the second most deadly disease human have ever discovered. Right? Even in retrospect, Peter described his own actions as an incredibly perilous act. Right. So after they opened the thermos, they found what appeared to be a soup of half-melted ice. So basically, when you're delivering medical samples, to prevent the samples from spoiling or denaturing, you're supposed to keep the temperatures below zero, right? However, because of, airplane turbulence, poor packaging and whatnot, the thermos, the cooling liquid in the thermos was everywhere, and several of the test tubes containing Ebola Zaire blood samples, which they did not know was Ebola Zaire blood samples, was now a soup of ice and of icy water and infected blood. Only one test tube was intact, and there was a handwritten note from a certain Dr. Jacques Cortelli, a Belgian physician who was working in a clinic in Kinshasa. And what he described was that the thermos contained 2 vials, each have 5 millimeters of clotted blood from a Flemish nun who was too ill to be evacuated out of Zaire. So, Flemish is a group of people, and a nun is a person who was working in a church, a woman who's working in a church. And when we talk about evacuating from Zaire, because she's Flemish, she's obviously not local African. So when there's a pandemic of sorts, like what happened with COVID-19, countries evacuate their own citizens out of the affected area. But she, of course, was too sick to be evacuated because Ebola hemorrhagic fever hits her body like a wrecking truck. So what they thought was she was suffering from a mysterious epidemic that has so far evaded detection because they didn't really know what it is. They still think it was yellow fever, right? So right now, Peter was very excited because he thought he was finally going to see a disease never seen before. Like, it was finally going to be a new disease, right? It's not going to be something like Lassa fever where people discovered it, then realized it was kind of, benign. I mean, not benign, but it wasn't very, it wasn't very dangerous. So, of course, he thought that this was his chance to go on an adventure, right? To go to Africa. This was his hopes and dreams, to go to Africa to identify new diseases, right? For Peter, when he was in university and he was talking to his professor about his future prospects in infectious diseases, his professor told him to drop it. Because his professor, along with a lot of the medical community, believed that infectious disease study was not a subject of utmost importance because most of the diseases had been discovered and had been conquered. And the ones that have been newly discovered recently, like Lassa fever in 1969, were not that, newsworthy. It's not going to be something like COVID, which has ridiculously high transmission rates. It's not going to be something like Ebola, which has ridiculously high death rate, right? However, he still wanted to go to Africa, and he still wanted to save lives, and still want to solve unsolved scientific questions. And that is why Peter still chose to work in infectious disease study. So now, Peter's lab opened the blood sample, and they tested it using this thing called Vero Cell lines. So when you're testing blood samples for whether it's infectious or not, you need to see if the blood sample contained any pathogens that can kill cells. So if you remember all the way back when viruses were first proven to exist, the scientist at the time, he took leaf samples from tobacco plants that were infected by the spotted leaf disease, purified it so that no bacteria could remain, and injected it into healthy leaves, and saw those healthy leaves get infected by the disease, thus proving there was a virus. So this principle is still in use today, where you try to get the sample, whatever the pathogen is the sample, to infect healthy cells to prove that these the sample contains pathogens that are deadly and infectious. For bacteria, you would usually just inject them into animals, alright? But for viruses, you do this thing called Vero cell lines. VERO. So what Vero cell lines are, they're basically rapidly multiplying cells. And modern day, if a lab wanted to conduct an experiment using these kind of cells, they would go and purchase them. However, back in the 1970s, you had to make them, prepare them yourself in the lab. And so that's what they did, right? Glass, not plastic petri dishes. Plastic was expensive back then, right? So they prepared the Vero cell lines, they injected the blood samples in, and they put the sample into the brains of some mice as well. And after a bit, happened was the Vero cell lines died. Like the cells on the Vero died, right? And when they died, they detached from the glass side of the containers, right? And the mice they injected the blood samples into also died. So that means they have identified that there is definitely a infectious pathogen inside the blood sample that they received from Zaire. However, just as they were beginning to grow the second Vero cell line, his boss, a man called Petri, stopped them, right? Because they had received instructions from the WHO, or World Health Organization, or WHO's Viral Disease Unit, to shape all of their samples to the biological materials from this new pandemic to Porton Down in Britain. Why Porton Down in Britain? Was because back then they felt like there was a chance that this disease was not your regular disease. It was going to be something kind of dangerous. It was going to be hemorrhagic fever, right? So that's what people were thinking, that this was going to be hemorrhagic fever because there's so many hemorrhagic symptoms of the people infected, so obviously it's hemorrhagic fever. And back in the 1970s, not every single biological lab is equipped with the right equipment and the right database or doctors or knowledge to handle hemorrhagic viruses. Back then, the Soviet Union was still around, and outside of the Soviet Union, only 3 of the labs could handle hemorrhagic viruses. One of them was in Porton Down in Britain, and even when they send it to Porton Down, Britain, they send it on to Atlanta. Atlanta has the headquarters for the CDC, which is the Centers for Disease Control and Prevention of the United States, and Atlanta has the best hemorrhagic fever lab. They're called the world's reference lab for hemorrhagic viruses. Of course, Peter and his boss Peyton, they weren't very happy with shipping all of these to Atlanta, right? With 2 other places. Why should we ship it to them when we were the ones who received the samples and we were doing this work, right? Why should we do this? So, as a result of that, they decided to be a bit rebellious. They kept some material back under the ruse that they were still busy packaging it, right? And they tested it. How to test it? They took the Vero cell lines. Now they've died, right? The first Vero cell line died, and now they take the ones that have died, and they need to look at it under electromagnetic microscope to see if there's any viruses. And when doing that, his boss dropped one of these test tubes, breaking all its extremely dangerous content on the floor. And back then, they managed to clear it up and nothing really bad happened, right? However, when they looked at it under the electromagnetic microscope, with the help of his boss's friend, a person called Wim Jacob, right? They saw something that was very weird, because they saw photographs of what appeared to look like Marburg. Now, for those who don't know what Marburg is another hemorrhagic fever with a death rate about 25%, and it appeared in Germany in a shipment of monkeys from Africa, infecting the workers that handled the monkeys, causing death in many of them. And yes, Marburg was a hemorrhagic fever. And why do we say Marburg looks like Ebola? Was because actually Marburg and Ebola both belong to the same family of viruses called a filovirus. And we'll explain characteristics of a filovirus very soon. So anyway, back to our story. So after they saw the photographs, Peyton, his boss, was not very happy. He was like, this looks like Marburg. However, Peter didn't know anything about virus that was known to be that long. It was 14,000 nanometers long, or about 0.000014 millimeters long. Might not sound very long to you, but for a virus, that is very long. Let me give you a comparison. Polio is 50 nanometers long. The polio virus, which is a virus that, famously Franklin Roosevelt, the American president, had it, that was only 50 nanometers. And then you look at Marburg, which is 14,000 nanometers, right? And so, what they thought was that, Marburg is a scary illness. And how do they test for Marburg? They can't really know for sure if they're testing, if they have got Marburg here, or if they just have a virus that looks very similar, right? And back then, to treat this kind of disease, you need Marburg-specific antibodies extracted from Marburg patients that recovered. They didn't have any of that. So, not being suicidal, they established that their virus was a very close relative of the most terrifying Marburg virus. They sent all of their remaining samples to CDC. However, they still felt unsatisfied. Peter felt that his long childhood fantasy about exploration was within reach, and that he should go to Zaire. However, they didn't have money. This is a very common theme when it comes to scientific research. You don't have money. So what they had to do was they had to run around trying to look for funds, and they could not really find funds. For example, he went to the Ministerial Department for Development Aid, and they said, we help programs to help poor people, not programs to assist in medical research. Even though assisting in medical research can help poor people, but people's brains are very interesting. So anyway, in the American labs, they also established that the virus was new. It was not Marburg, but it was very similar to Marburg. And the person who told them this news was a person called Carl Johnson, who was the chief of special pathogens at the CDC. So now, are they going to actually go to Zaire? Well, maybe not, right? So right now, Peter was very crestfallen because he felt that the segment was over. He's going to go back to his regular Monday routine of looking at, stool samples of patients with non-specific belly aches to figure out what bacteria they had. However, on October 15th, right? Peter and his wife, Greta, went to Paris, right? They went to Paris for a weekend. However, they got an urgent call saying that they should contact the phone number in Brussels. So they were like, why should I do that? So he called his boss, who was still at the lab, and he said that the Department for Development Aid and Minister of Foreign Affairs want to meet him. Why? Because they want to go to Kinshasa. Why do they want to Kinshasa? Because Belgium had relationships to Zaire. They were previously, a colonial force in Zaire. So, as a result of that, they need to do something. And also, people in Belgium have been panicking, right? So that is why the Belgian government is now, under pressure to do something, right? Peter was like, why me? I'm just like 20-something. I'm just like a recently graduated medical student. Why should I go to Zaire? Right? But however, he didn't want to go. He didn't protest, right? So, he goes to Africa. And that is how Peter ended up in Africa. When he went to Africa, he had many other adventures. He did some ground research on the disease of Ebola. He participated in the naming of the disease of Ebola. And after that, he even lived for a long time inside a very poor region of Zaire that was hit hard by the pandemic. However, that is content for next time. Stay tuned for the second session of our Ebola class, and if you have any inquiries, you can leave them in the comments, and you are also free to leave any improvements and suggestions you have on my course and my videos in the comments below. Thank you for watching. After listening to such an inspiration talk on Ebola in Africa, do you feel incentivized to do something about the poverty in Africa yourself? Well, if you are in luck. The charity organization Shining Horizon AMPM is setting out on a mission to solve malnutrition in the country of Eritrea in Africa, where 66% of people live below the poverty line, and the level of malnutrition in the country is at a crisis level according to the WHO. If you want to make a difference, please donate to Tencent Charity and support our mission in providing the children of Eritrea with adequate nutrients. That is Shining Horizon AMPM, solving malnutrition in Eritrea. Thank you for your support and donations.